Hello, my name is Caprici Wallace and this is cancer, an illness of the cell cycle. Cancer is a result of unregulated cell division, so in order for us to truly understand what causes cancer, we have to understand the cell cycle. Let's start with the first and the longest phase of cell division, interphase. Interphase is up about 90% of cell division and it is basically the period of growth. That period of growth consists of three subphases. First is G1 or grow 1. This is when the cell grows and expands by producing proteins and replicating organelles to prepare for cell division. Next is S or synthesis. This is when the chromosomes in the nucleus are copied into two sister chromatids. The sister chromatids are connected by centromeres, which is shown by the green dot. Remember, the cell is still growing throughout this process. Next is G2 or grow 2. The cell is growing to prepare for mitosis. As you can see here, mitosis really only takes up a small portion of the cell cycle. Regulation of the cell cycle is important. So important that the cell cycle has signal molecules that check for errors in DNA at certain checkpoints. Signal molecules carry out checkpoints to ensure that each phase is being carried out completely and accurately. If an error is discovered at one of the checkpoints, repairment is attempted, but if this fails, the cell enters G0 phase, where it does not divide at all. The G1 checkpoint is the most critical, being that it's the first of them all. It takes place towards the end of the G1 phase and it analyzes the DNA. When an error in the DNA is discovered, then the DNA repair enzymes attempt to fix it. However, if the error can't be fixed, then the cell undergoes apoptosis, which is just a fancy way of saying cell death. The G2 checkpoint takes place towards the end of the G2 phase. This analyzes DNA and determines whether or not it's ready to undergo mitosis. The M checkpoint takes place during mitosis, during metaphase. This checks to see if the microtubules are attached to the chromosome properly. And I always remember the function of the M checkpoint by remembering M and M. Cyclins are proteins that activate or turn on cyclin-dependent kinases, or CDK. Cyclin-dependent kinases are enzymes that help advance the cell past each of the checkpoints in the cell cycle. This is an activated CDK. We can also say this is a mitosis promoting factor, which is an activated CDK. So a cyclin attached to a CDK. A mitosis promoting factor helps advance the cell cycle to mitosis, or it promotes the cell to mitosis. Platelet-derived growth factors are signal molecules that are released by the cell, and when they reach a receptor of a damaged cell, they catalyze cell division. So cancer is a result of disturbances in the cell cycle. Normal cells have density-dependent inhibition, which is when cells sense that there is no more room to divide, so they don't. Cells that are cancerous don't have that inhibition. After a normal cell is transformed into a cancerous cell, that cell, if it isn't destroyed, can evade checkpoints and grow and divide uncontrollably. It will continue to divide in spite of crowded cells. Eventually, that growth will spread into other tissues and organs and damage them, and possibly eventually kill the organism. And that's the cell cycle, that's cancer and how it happens, and I hope that was helpful.